My name is Joshua Mugabe. I welcome you to Coordination, and we are going to look at the learning outcomes. But we shall start with examining the role of plant hormones in tropism, photoperiodism, and the application of these processes in agricultural practices. We shall be looking at all the learning activities, but we shall start with the activity A, which says that in groups, learners design and carry out scientific investigation on phototropism and geotropism. We have item 1, which is going to be on phototropism. Your class is given a tray of young bean seedlings. The teacher covers one side of each tray, so light comes from the left only. Task 1. Predict what will happen to shoot growth over 72 hours. 2. Explain the hormonal mechanism. Which hormone, how it moves, what it causes. 3. Suggest one agricultural situation where this knowledge is used. Let's start with task 1, which says predict what will happen to shoot growth over 72 hours. In our response, the shoot will bend toward the light, and that is what we call positive phototropism. Task 2, explain the hormonal mechanism. Which hormone, how it moves, what it causes. The mechanism, light causes photoreceptors, which are phototropins, on the illuminated side to trigger redistribution of oxygen to the shaded side of the shoot. Higher oxygen on the shaded side stimulates cell elongation, there causing the shoot to bend towards the light. Task 3. Suggest one agricultural situation where this knowledge is useful. Application. In greenhouse seedling production, uniform overhead lighting prevents seedlings from bending and becoming weak. Growers use even lighting or supplemental lights to produce straight stems for transplanting. Item 2, it is on phototropism. A student notes bean seedlings in a pot, all leaning toward the window. The task. Explain the physiological mechanism and the name of hormone involved. Seedlings show positive phototropism. Cells on the shaded side elongate more because oxygen redistributes to the shaded side, promoting cell elongation there. This makes the shoot bend towards the light source. Item 1 on geotropism. You plant seeds in 3 containers. A. Seeds on top of soil. B. Seeds on side of narrow transparent tube. C. Seeds on inverted lid. You observe root and shoot orientation. The tasks. Predict root and shoot orientation in each container. Explain the sensory mechanism and hormonal prayers. Give one agricultural implication. Let's start with predict root and shoot orientation in each container. Roots will grow downward, that is positive geotropism, and the shoots upward, which is negative geotropism. In all but constrained cases, tubes may force curvature. Explain the sensory mechanism and hormonal prayers. Statolith, those are starch grains in root cap cells, settle under gravity, signaling oxygen distribution and growth adjustments. Auxins inhibit root cell elongation on the lower side, so it curves downwards, and stimulates shoot elongation on the lower side, so shoots curve upwards. Three, give one agricultural implication. In containerized horticulture, root orientation affects transplant success. Containers and planting depth are managed to encourage proper root systems. Let's look at item 2, and this one is about geotropism. A farmer plants a potato tuber on its side. After sprouting, shoots curve upward and roots downwards. Task: explain using hormones. Gravity is sensed by statoliths in root and shoot cells. Auxins become more concentrated on the lower side. In shoots, this promotes elongation, so they bend up, that is negative geotropism. In roots, higher auxin inhibit elongation, so they bend down, and that is positive geotropism. Item 1, still on photoperiodism. A greenhouse manager wants a short-day Silsathium to flower area. What can they do? For short-day plants, long nights are required. The manager must avoid night interruptions by light. To induce flowering area, they can shorten the photoperiod, that is, increase continuous dark, or manipulate phytochrome state, that is, use far red light at appropriate times is risky. Alternatively, apply gibberellins if the species responds, but manipulating night length is primary. We want now to carry out an investigation 1 on phototropism in young seedlings. And in this investigation, our aim is to investigate how unilateral light causes curvature in shoot tips and test the role of the tip and auxin transport. This one takes us to our hypotheses. We can have very many hypotheses. The first one, seedling shoots exposed to unilateral light will bend towards the light. That is positive phototropism. The second hypothesis, removing or shading the shoot tip will prevent the phototropic response. That is, the tip is required for perception. The third hypothesis, blocking oxygen transport will reduce or eliminate bending. The materials, we shall need beans or oat seeds, the first germinating, at least 30 seedlings. Seedling pots or damp paper towels in trays. 2 light sources, lamp with adjustable position, or 1 lamp and a dark room. Opaque cups or small aluminum foil cups to cover the shoot tips. A ruler, protractor, camera or phone for time-lapse photos. And then we shall also need labels, markers, and notebook. Experimental design and variables. We shall need groups of seedling, 8 to 10 seedlings per group. 1. Control. Light above even illumination. Unilateral light. Light from one side. Tip covered. Unilateral light, but shoot tip covered with opaque cap. Oxygen transport blocker and unilateral light plus NPA applied near the tip if possible. The independent variable, we shall have light direction or tip treatment. Dependent variable, that will be the angle of curvature of the shoot or the degrees. This one is measured from vertical after set time. For example, 6, 12, 24, or 48 hours. Controlled variables, we shall use seed age, temperature, moisture, light intensity, and the species. Methods, step by step. 1. Germinate seeds until coleoptile or first true leaves appear, and that would be around 2 to 5 days depending on the species. Place seedlings vertically in trays. For the unilateral group, place a lamp approximately 20 to 30 centimeters to one side for control. Place lamp above. At time O, mark initial orientation or photograph. Start the timer. For the tip covered group, carefully place an opaque cap over the shoot tip without damaging it. For oxygen transport blocker group, apply NPA solution around the coleoptile. Best following safety directions. Alternatively, use lanolin paste with and without IAA for manipulation experiments. Measure curvature angles at 6, 12, 24, 48 hours using a protractor or image analysis of photos. Record data. Sample data table example. We have the seedlings ID 121212, control, and the hour after 6 hours, 12 hours, 24, 48. Control 1, 5645. Control 2, 4565. Unilateral 12, 25, 40, 55. Unilateral 2, 10, 22, 38, 50. 1. Tip covered 3, 2, 1, 2. 1. NPA 4, 6, 8, 10. Expected results and interpretation. Unilateral light, progressive increase in curvature toward light, the largest angle. Tip covered, little or no curvature, shows tip needed, site of perception and oxygen redistribution. Oxygen transport blocker, compromised curvature, demonstrates role of oxygen transport. Conclusion supports oxygen redistribution from tip, causing cell elongation on shaded side. Investigation 2. We're going to deal with geotropism in roots and shoots. The aim of our investigation: compare shoot versus root responses to gravity and test whether clinorotation, that is rotation a seedling slowly, eliminates directional growth. This takes us to our hypothesis. 1. Roots will show positive geotropism, that is, grow downwards, and shoots negative geotropism, that is, growing upwards. The second hypothesis: placing seedlings on a clinostat rotating platform will reduce directional growth because gravity vector is averaged. Our materials. We shall use germinated seedlings. Those are beans, peas, at least 24. Transparent jars or petri dish so you can observe roots. Clinostat or a simple rotating turntable. Slow rotation to average gravity. Labels, rulers, camera, protractor, and we shall need supports to hold seedlings horizontally. Experimental design and variables. Groups we shall have 8 per group. Vertical control. These are normal vertical orientation. Horizontal placement. Seedlings placed horizontally. Observe bending. Horizontal plus clinostat rotation. The seedlings on a clinostat rotating slowly, approximately 2 to 6 rounds per minute, or much slower for seedling safety. Independent variable, that will be orientation and rotation. Dependent variables, we shall have angles of curvature of roots and shoots, root growth direction after 24 to 72 hours. The methods. Germinate seedlings so that roots are visible, then transfer to transparent container and mark initial orientation. For horizontal group, lay the container so that seedling axis is horizontal. For clinostat group, mount container on clinostat and start slow rotation continuously. Observe and measure root and shoot angle after 12, 24, 48 hours. Expected results. Horizontal, the non-rotating, shoots curve upwards, roots curve downward over time. Clinostat, rotating, direction of bending is reduced. Roots and shoot grow more randomly or helically because gravity vector is averaged out. Showing gravity perception is directional and requires a consistent vector. Let us now have an ecological and agricultural significance report. Our title: The Role of Plant Hormones in Tropism and Photoperiodism with Ecological and Agricultural Implication. Our introduction. Plants continually adapt growth and development to the environment. Tropism, that is phototropism and geotropism, a directional growth response mediated principally by plant hormone auxin, while photoperiodism, that is day-length responses, involve photoreceptors, the phytochromes and cryptochromes, interacting with the circadian clock to regulate the production of flowering signal such as frolicin and the action of hormones like gibberellins. This report investigates phototropism and geotropism experimentally and discusses ecological and agricultural significance. The methods. 2 experiments were performed using bean seeds. Phototropism, seedlings exposed to unilateral light, tip covered and auxin transport inhibited groups, curvature measured at intervals. In geotropism, Seedlings placed horizontally or on a clinostat, root and shoot curvature measured. Results. Unilateral light induced significant shoot curvature towards the light. Tip-covered seedlings and those with oxygen transport blocked showed greatly reduced curvature. Horizontal placement induced shoots to curve upward and roots downwards. Clinostat rotation minimized dilated bending. Sample data show mean shoot curvature in unilateral light group reached approximately 50 degrees by 48 hours, while tip covered remained less than 5 degrees. Discussion and methods. Phototropism mechanism. Light perception in shoot tip triggers asymmetric oxygen distribution to the shaded side. Increased oxygen there stimulates cell elongation, causing bending towards light. In geotropism mechanism, statolith settling in columella root and similar gravity-sensitive cells in shoots lead to uneven oxygen distribution because roots and shoots differ in sensitivity. Oxygen stimulates shoot elongation but inhibit root cell elongation. Photoperiodism. Leaf photoreceptors detect day length. The Scandian system gates FT stop fluorescent production. FT moves to apex to induce reproductive development. Gibberellins can modulate flowering in some species. Ecological significance. Tropism helps seedling orient for optimal light capture and anchor roots for water nutrient uptake, crucial for survival and competition. Roots geotropism ensures efficient soil exploration and stability. Photoperiodism synchronizes life cycles, flowering, dormancy, with the seasonal environmental conditions, promoting pollination success and seed set in favorable conditions. Agriculture significance and application. Phototropism. Growers use light direction in nurseries and greenhouses to train plants, reduce shading, and optimize canopy light distribution. Understanding oxygen allows use of rooting hormones, IAA, IBA, to encourage root formation in cuttings. Geotropism. Knowledge of root orientation helps in transplanting and mechanized sowing. Plant breeding for root angle affects nutrient capture and drought tolerance. Photoperiodism. Controlling day length in greenhouses forces flowering out of season. In floriculture, chrysanthemums, poinsettia, artificial lighting schedules, and blackout curtains are widely used. Common use: synthetic auxins and gibberellins are used as plant growth regulator to enhance rooting, prevent premature fruit drop, control height with growth retards, or induce parthenocarpy. However, responsible use is necessary to avoid residues and unwanted growth patterns. Conclusion: auxin-mediated tropism and photoperiodic hormone signaling are central to plant development. Laboratory investigations confirm the requirement of tips and oxygen transport for photoperiotropism and show the direction role of gravity in shaping roots and shoots. In agriculture, these mechanisms are exploited to control flowering time, improve propagation success, and optimize plant architecture for yield and management. Recommendation for students and farmers. For classroom, repeat phototropism experiment with and without tip covering and create time-lapse videos. Powerful demonstration. For farmers, use photoperiod control and appropriate PDRs only in line with extension, advice, and local regulations. Consider root angle traits in variety selection for drought-prone fields. Revision. Tropism. This is a directional growth response to environmental stimuli. That is light, that is phototropism, gravity, geotropism, or gravitropism. Main hormones: auxin, indole 3-acetic acid, IAA. Auxin is produced in the shoot tips and redistributed asymmetrically when stimulus present. Phototropism in shoots. Unilateral light results in auxin moves to the shaded side of the shoot. That causes higher auxin there, resulting in cell elongation on the shaded side, and the shoot bends towards light. Geotropism in roots and shoots. Gravity causes statolith, the starch grains, to settle in cells, causing oxygen redistribution in shoots, more oxygen in the lower side, cell elongation, shoot bends up. In roots, high oxygen inhibits cell elongation. The lower side grows less. The root bends down. Other hormones. Cytokinins and gibberellins modulate growth rates. Ethylene can interact, that is, example, triple response in seedlings. Auxin and cytokinin balance affects organogenesis. Photoperiodism. Plant developmental responses, especially flowering, to the relative length of light and dark period. Light receptors and signals. The phytochromes, red and far red, and cryptochromes, blue light, measure their length. The photoreceptor state interacts with the plant circadian clock to determine if flowering signal is produced. Hormonal molecular pairs. Production of florigen, protein in many species, is regulated by photoperiod and transported from leaves to shoot apical meristem to induce flowering. Gibberellins can promote flowering in some long day and biennial species. Auxins and cytokinins affect overall growth that interacts with flowering. Agricultural use. Manipulating photoperiod light and dark cycle using growth regulators, GA, PGA, RS, controlled environmental agriculture to force or delay flowering and fruiting. Thank you for participating with us in coordination and the learning outcome. Examine the role of plant hormones in tropism, phototropism, and application of these processes in agricultural practices. We have been looking at learning activity A, whereby in groups learners are to design and carry out scientific investigation on phototropism and geotropism. So let us find ourselves in the next learning activity. Please, if it has been useful to you, you can subscribe and please come back again. Thank you.